All right, so welcome everybody. I'm really so pleased that everybody, that this is taking place. You know, this is the first time. I'm not sure, I'm assuming most of you are familiar with CanVECTOR, but it's a national venous thrombosis sort of consortium that was started, must be 8 or 9 years ago already. It's primarily clinical, but we basic scientists sort of needled our way in to create more translational opportunities and some communication between the basic scientists and the clinicians. And they've been fantastic in supporting this. And as a consequence, CanVECTOR has raised various funds from a whole bunch of donors. And let me, not try to hit the wrong key. I'm going to go back to that. A bunch of donors, as you can see, they don't all support the basic scientists. I wish they did, but they do support, and we've been able to have varying support for students at varying levels to do basic research in hemostasis and thrombosis, I would say. And it's really bringing a community of Canadians. We Canadians have a long history of clinical and basic science in the field of thrombosis with many luminaries, and I won't try to name them because I'll forget people. So today, and I should thank Paul. Paul, where are you, Paul? Paul is my colleague in arms. He's technically on leave, taking care of child. But Jesse and the rest of the CanVECTOR team has really led the way on this, and the students, of course, they've been driving this. So those were my welcome and opening remarks. We're going to have trainee presentations, and I'm encouraging people to keep their once the presentations are done, to open up your videos and ask questions directly. You can send them in by chat. You're more than welcome, but I'd really encourage you to make this a little freewheeling and a little more convivial that way. And then we're really honored to have Valerie Tutwiler, who's going to give a keynote presentation, and then we'll have some awards. Now, speaking of awards, how are we doing? Where's my slide on the awards? Oh, here we go. All right, Jesse, do you want to describe this? Because I have no clue.
Oh, sure. We wanted to have a way for people to vote on the presentations that our trainees are going to give it a little competition feel, and of course, to give them a little bit of feedback. So the presentations are going to be relatively short, but we did put together some criteria that are on this slide here that you can see that, of course, the trainees have received in advance so that they can tailor their presentations for it. And in order to vote, if you can on your browser, open up the link that I posted in the chat, or you can copy paste it into your browser, or you can go to Slido and enter that number that I have on top of the slide here, 616890. And you'll be able to just rank the presentations as they go, keep it open, and you of course submit after all of the presentations are done. So you only submit one time based on how you're ranking all those presentations.
Any questions? Everybody's ready to go? Are all the speakers ready and okay? Do they have access to share their slides?
Yeah, once you stop sharing your screen, the next one will be able to start.
Okay, so I'm going to start. I'm going to introduce the first speaker is Amin Zarai. I apologize for incorrectly bad pronunciation of names. I should have.
That was spot on, actually. No worries at all.
Okay, but I do usually get ready. Okay, he's from the University of Ottawa. Supervisors are Dr. Delic and Carrier, and I will let him take it over. All right, I'm going to stop sharing.
All right, I'm just going to quickly share my screen here. Can everyone see my slides?
Yes, we can share them. If you just want to move to presentation mode, then we'll see just the slide.
Yes. And do you, Jesse, do you also see my scroller?
I can see your cursor now that it's on the slide, but
okay,
I'm sure that'll go away in a second. But this looks great.
Okay, awesome. Thank you so much. Once again, well, thank you to everyone, first of all, for organizing this event, and congrats to all the studentship awardees on this fantastic award, and thank you to CanVECTOR for providing this opportunity. I've personally been very fortunate to be supervised by Dr. Carrier and Dr. DeLuc, both are physicians at the Ottawa Hospital, and they're hematologists, and that's sort of going to be what my presentation is going to be about. So treatment of splanchnic vein thrombosis with anticoagulant therapy in cancer patients, and it's going to be a systematic review and a meta-analysis. So I'm just going to provide a sort of a layout for today's presentation just to get the ground running here. First, going to be presenting a little bit of background on splanchnic vein thromboses and their current treatment in cancer patients. Then going to study objectives and aims, and then a little bit of my methods, results, and conclusions at the very end. So I kept saying splanchnic vein thrombosis, and definitely a bunch of confused faces. So what exactly is this? Really, blood clots usually are, these are called venous thromboembolisms. They're normally in the, either in the deep veins of the arms or legs or in the lungs arteries. And these splanchnic vein thromboses, they're sort of unusual because of the site of the clot. And it really happens in the splanchnic venous circulation that drains the digestive system. And if these unusual clots expand, they may block and sort of block the blood supply to key digestive and accessory organs, causing mesenteric ischemia and potentially death. And the incidence of these sort of unusual clots is much lower than the usual side ones, 25 times lower, but there's a lot of variability in the reporting of these more unusual clots in terms of their incidence, site, and type in different patients. Now, cancer patients, they generally tend to have cancer, is a fairly big risk factor for patients for blood clots, and there's an increased risk of recurrence of VTE in these patients after they get anticoagulation, meaning that you know, the blood clot can actually come back once it's treated with anticoagulation in these patients. They have a higher risk of that, and the treatment of specifically these unusual site thromboses in cancer patients is fairly challenging, again, because of the heterogeneity of the population of these SVT patients. And at the same time, guidelines and sort of literature that's available for effective treatment for these unusual clots is fairly limited, and it mostly comes from expert recommendations and small observational studies, and it's often not really specific for cancer-associated SVTs. However, there's been some cohort studies, some observational studies recently that suggest that anticoagulation might be effective and safe in most SVT patients, but it's, again, not specific for cancer patients. There's also been an ISTH guidance document suggesting that low molecular weight heparin and DOACs, which are a type of anticoagulation, might be useful for patients with cancer and SVTs. And that's sort of what my presentation is going to be about. It's sort of just determining, you know, the treatment type, the duration, the dosing, and what populations of cancer patients would be benefiting from what sort of a treatment of anticoagulation, and then if what's the safety parameters of this. And because these patients tend to have a unknown risk of bleeding after anticoagulation. So really my aim is to determine the type of anticoagulation, duration, dosing, effectiveness, and safety in adult patients with cancer-associated splanchnic vein thrombosis. Very difficult word to pronounce. I always try to avoid it. And then to also assess for outcomes such as SVT resolution, recurrence of the clot, vein recanalization, progression, bleeding, and mortality after anticoagulation. So how did I do this? Really, systematic reviews are just you know, you want to gather as much recent clinical evidence as you can on the topic that you're doing. And the way that you do it is you come up with a specific search strategy, and you search through, in my case, clinical databases. So I looked at Embase, Medline, Central, and clinicaltrials.gov, and looked for any studies that evaluated really the use of anticoagulation to treat patients with cancer-associated SVTs, the adult patients, and compared really patients who received anticoagulation to those who did not. And also, I was interested in looking whether specific subgroups of cancer patients, for example, those who have solid cancer compared to those who have more hematologic or blood-related cancers, called MPN, would benefit from any particular type of anticoagulation treatment. And then I used the Robbins-I tool to assess the risk of bias in observational studies. So just a quick rundown of what I found. I have on the right the PRISMA document suggesting, you know, the flow of how many studies I found and how many were included. And There were really 18 studies that we found, and 6 of them were abstracts, 12 of them were full text. And this really gives you just sort of an introduction of the scarcity of the resources that are available on this topic. And 8 of these 18 were actually in solid cancer patients only, and 10 of them focused on these more hematologic cancers. And only 4 of the total 18 looked at provided baseline characteristics for both patients who received anticoagulation and those who didn't. But if we look at the duration of anticoagulation and really the range that was provided in these studies, we can say that duration ranged anywhere from 3 to 12 months, and it was fairly variable, and it was difficult to really do any meta-analysis on this. And then the type of anticoagulation that was used in these studies ranged from use of low molecular weight heparin and VKAs to VKAs, and then to DOACs and a combination of those, and then heparin, and 2 of them didn't actually mention, unfortunately, the type of anticoagulation. So what exactly did I find after all of this work here? It really, as I mentioned, the 2 subgroups that I wanted to look at were solid cancer patients and MPN patients, these hematologic cancer patients. And in solid cancer patients, what you see in front of you is the meta-analysis for VTE recurrence. So if the more we go to the right here from that so there's a x-axis there, and where the mark for the 1 is, that's sort of a statistical insignificant margin, if you will. And if any of the ranges of the results passes that would mean that there's no statistical significance. And if you look at the results here, actually for VTE recurrence, anticoagulation really didn't differ. Patients who received anticoagulation really didn't have a different outcome for VTE recurrence compared to those who did not receive anticoagulation. And what I'm, by the way, presenting here is the incidence rate ratio, the IRR that you see there. And then the same thing when I looked at bleeding, and this includes all bleeding types, we sort of accumulated them together. We noticed that there's a little bit of a tendency for anticoagulation to actually protect against bleeding in solid cancer patients, because the incidence rate ratio was actually much higher, a little bit higher than 1, although there's like a slight overlap with 1 as well. And for mortality, there wasn't really any benefits because anticoagulation wasn't able to help these patients, unfortunately, with solid cancer patients. And however, when you look at the MPN patients, there is, in effect, there's a benefit for patients there for taking anticoagulation. And you can see that there's like about a 51% risk reduction compared to those who are not taking anticoagulation. And this is very good for us because, as I said, there's a scarcity of data. And if you look at the number of studies that we've included, there's only 3, Di Stefano, Greenfield, and Tremblay, just to give folks a little bit of a idea there. And also the heterogeneity of the results were interestingly very, very low. You can see that it wasn't statistically significant. The P was 0.94, just to the bottom left there. But nonetheless, it's interesting to know that there's a sort of a protective effect against VTE recurrence in these MPN patients. And when you look at the bleeding, we saw that there's no difference between these 2 patients, although it's leaning a little bit towards favoring anticoagulation for bleeding there. And then, as I mentioned, there's bias in all studies. It's inherent in their design, and specifically for us, we weren't able to find any randomized controlled trials. They were all just observational studies and cohort studies. And if you look, really, I have to the right here, which I'm not going to even go into. It's quite a grid that I've created, but it really is just representing the risk of bias. So if it's a white circle, it's no information. We couldn't find any information there. And if it's low risk of bias, moderate, serious, and critical, and most of the studies that we included out of the 18 were of serious risk of bias, or they had a moderate risk of bias. And you can really see that, unfortunately, the results tend to be biased, and this is mostly because of confounding factors that's inherent in the design of these more observational studies compared to non-randomized studies. So just, what's next really in line, and what's the conclusion that we can grab from this? Again, meta-analysis from this study only included observational studies. There wasn't any RCTs, and because of the serious risk of bias, because of confounding an indication, we really can't draw firm conclusions for now. And there was a scarcity of specific anticoagulation regimens, so we couldn't really, do any subgroup analyses based on the type of anticoagulation or, unfortunately, the duration of anticoagulation. And this is because not enough studies had enough data for us to be able to do that. But anticoagulation, as I said, appears to be fairly beneficial in reducing recurrence of VTE at 12 months in MPN patients, which is a really nice finding. We were fortunate enough to be able to do. But there's no increased risk of bleeding, which is also important because physicians need to really balance that, right? They need to balance whether the clot has gone away, that it's not coming back, and whether there's not an increased risk of bleeding for these patients. For solid cancer, it was the opposite, where there wasn't any statistical effect on recurrence of VTE, but it was a little bit worse on anticoagulation for bleeding. And just overall, this topic, we of course came up with it through discussion with experts, and of course my investigators are one of them is a world leader in cancer-associated thrombosis management. So we find that it's fairly relevant, and we were happy to be able to find some benefit for MPN patients. Solid cancer patients, what we think might be the case of why we didn't find any effects is perhaps because of first of all, the low amount of studies, again, that we weren't able to find. And some of the outcomes, some of the studies didn't even report outcomes, unfortunately. So we definitely, there's a large need for RCTs and sort of these higher level study types. So with that, I just wanted to sincerely thank my investigators, first of all, Dr. Carey, Dr. DeLuc, for their unwavering support, and the Ottawa Blood Disease Center and the Ottawa Hospital, the University of Ottawa, and of course, the CanVECTOR's training, mentoring, and early career development platform for us trainees. It's wonderful to be able to be sort of supported with an award to support our research initiatives. And so with that, I just thank you very much for listening, and would like to entertain any questions. I see that
Thanks very much, Amin. Can you turn off your slides?
Yes,
Amin,
absolutely.
that'll be good. Then we can see everybody, hopefully. And we are open to questions. Anybody? You can put them in
I
the
think
chat.
Allison has her hand up. Go ahead, Allison.
Just go for it. Yeah, just
thanks,
jump in.
Amin. When I look at those forest plots,
Yeah.
the event rates were relatively low in most of the studies.
Yeah.
And you weren't able to pull out any sort of anticoagulants, but can you tell me in general what sort of were they using low molecular weight heparin? Were these patients put on unfractionated heparin for a period of time? What were they using for their anticoagulants in most of these studies?
That's a really, really great question, Allison. And actually what I can do is, Ed, if it's okay, I'm just going to share back my presentation just to make it a little bit easier to follow here. And I do have, I'm just going to exit presentation mode if possible. No, it's not. That's fine. The low molecular weight heparin and VKAs were the highest usage of anticoagulation across the studies that I found. And I think it really speaks to the fact that when physicians are not sure, when researchers are not sure, they just go with VKAs initially or low molecular weight heparins initially before moving into the more newer direct oral anticoagulants or whatnot. And after that, I believe the next highest on the list was VKAs, so vitamin K antagonist only. And then it went into the low molecular weight heparin, the VKA and DOACs. And there's more RCTs that are required for sure. And I had a discussion with Dr. Carey and Dr. DeLuc, and we're thinking of the next step for this to be that exactly, to propose a RCT regarding the comparison between low molecular weight heparin and DOACs, and I think it's going to be beneficial for patients and clinicians to have that data. But I think that's a really good question for sure. So,
okay, Ed, we're gonna have to cut questions short. So
We can cut
PDFs.
this short. I was wondering about the other part of disease. Did these papers report on the cancer progression? Because certainly, at least in mouse models, there's real solid evidence that tumor progression is affected by the other aspects of coagulation, cell signaling, angiogenesis, and so forth. Was there evidence of cancer progression?
That's a really good question, Ed. Unfortunately, not that much. So maybe there were, a few studies, but we really weren't collecting that, and we didn't put that as a protocol, although I think it would have been useful given the fact that it is probably involved with the response to anticoagulation, even for these patients. But no, not to my recollection, there weren't too many that noted remission or the progression in cancer. It was mostly just the thrombosis journey. So that's what we've been exploring. Yeah.
Thank you.
Oh, Ed, I think you're Ed Conway, sorry.
That's me. Yeah, I'm
muted,
muted. Yeah. Thank you very much, Amin. That was great. I think, okay. So the next speaker is Henry West from UBC, supervisors at Prydale. He's going to speak about tissue factor, some molecule that's somewhat interesting. Henry, you around?
Just a little bit interesting, yeah.
Okay, go for it.
All right. Hi, hello everyone. Like Ed mentioned, I'm Henry West, and I study at the CBR at UBC, supervised by Dr. Ed Prydale. And I'll be giving everyone a broad overview of one of my projects where we investigate one possible cause of the effects of viral infections on hemostasis. So if we search any coagulation or hemostasis and then almost any envelope virus infection, you will likely find some results. Herpes simplex virus, for example, is mainly thought of as causing cold sores, but there are many reports of its infection resulting in some hemostatic complications. HIV infection is another serious infection that is associated with effects on hemostasis, with one study finding that HIV positive individuals were 12 times more likely to suffer from early onset thrombosis. A little bit of an interesting one is dengue virus, which is mainly thought of as a hemorrhagic virus or one that causes bleeding. But it has been thought that, that this bleeding is a result of depletion in clotting factors, likely due to an excess activation of the clotting system, at least in part. And I mentioned these particular examples because these are viruses that the lab studies and some of which are involved in my project. And there could be a variety of causes for these observations, but the end result is still that coagulation and viral infection is closely linked. The Prydale lab has been developing a model of coagulation initiated on the viruses themselves. The viral envelope in these envelope viruses is derived from a virus's host cell's membrane. So when the virus buds off, it takes with it some of this membrane to form an envelope, and host proteins may still be attached to this membrane. Tissue factor is one such transmembrane protein and is a critical cofactor in the initiation of coagulation. So when it's released from the cell and residing on the virus surface, tissue factor no longer has its activity as tightly controlled and can freely act as a cofactor for factor 7A in its conversion from a factor 10 to factor 10A, which then downstream can result in the activation of clotting or of thrombin and result in plasma clotting. But tissue factor isn't only known for its procoagulant activity, it's also quite involved in cell signaling. And an extension of that coagulation model involves the cell signaling. So protease activated receptors are cellular receptors that are activated by proteolytic cleavage. In complex with tissue factor, factor 10A can cleave and activate PAR2, and thrombin can cleave and activate PAR1. And data from our lab has shown a greater infectivity of HSV1 or herpes simplex virus type 1 in cells stimulated with either thrombin or factor 10A. And this thrombin-based enhancement was inhibitable by blocking PAR1 activation, and the factor 10A-based enhancement was PAR2 inhibitable. And this isn't just an in vitro observation, but also holds in a mouse model of HSV1 as well, and suggests a possible role for tissue factor in the enhancement of infection in viruses. And many cells that are infected by envelope viruses express tissue factor. So we weren't surprised to have found tissue factor on the surface of every envelope viruses that we have examined thus far. And I've just shown a few here. And these are electron micrographs where gold conjugated antibodies were used to label viruses for tissue factor. dengue and HIV grown in and purified in the lab have been shown to contain tissue factor. But more interestingly, HIV derived from patient plasma, which has never touched cell culture, is also staining positive for tissue factor. And this shows that this isn't just an artifact from lab propagation. Taken together, the lab's broad hypothesis that guides my project is that the presence of tissue factor is common on all viruses and contributes to infection and pathologies associated with viral infections. And my main project aims to expand the number of viruses known to contain tissue factor, as well as provide possible explanations for these clotting events that are observed in infections in the cases of HIV and dengue virus. And one of the ways that we assess procoagulant activity is a clotting assay, where the virus is added to pooled plasma, and then calcium is added to initiate the reaction. Tissue factor activity on the virus will result in the activation of the coagulation cascade, and a reduction in clotting time means more tissue factor activity. And we also employ some inhibitors of tissue factor activity in this assay to demonstrate that tissue factor is the only cofactor accelerating the activation of factor 10. I completed pilot experiments with the clotting assay with both HIV and dengue virus. And with both viruses, as more virus is added, the faster clotting time is observed. And this makes sense if we remember that the virus is the only source of tissue factor. If no virus is added in both cases, the plasma does not clot before the cutoff for the experiment. The tissue factor inhibitor, NAPC2, prevented plasma from clotting in both cases. The other assay that we use commonly to show tissue factor procoagulant activity is a factor 10A generation assay, where the virus is mixed with factor 10, factor 7A, and calcium. And then using tissue factor as a cofactor, factor 7A can convert factor 10 to 10A, and we can follow this conversion through cleavage of a chromogenic substrate by 10A. The same inhibitors used in the clotting assay are also employed here. Mirroring the clotting assay results, there is a titratable effect as more virus is added, with more virus resulting in greater factor 10A generation. The results were shown to be dependent on the presence of a virus, or the virus, and the addition of inhibitors significantly reduced the activation of factor 10. And though preliminary, these and the clotting assay results still suggest a possible explanation for the altered hemostasis seen in during infection. So tissue factor is most known for its role in coagulation, but it also mediates inflammation and is a molecular link between these two processes. We've identified it on multiple viruses so far, and it's likely on many more. Its activity on these viruses provides a possible explanation for hemostatic effects observed during viral infection. And if its ability to promote infection, as observed in the context of HSV1, is demonstrated on other viruses, it may represent a broad spectrum antiviral target. My preliminary experiments in this project demonstrate tissue factor-like activity associated with HIV and dengue virus, and I'm in the process of confirming tissue factor antigen on the surface of these and other viruses. I'd like to thank the members of the Prystel Lab, as well as all of our collaborators and funding, without which this project and the ones that preceded it would not have been possible. And I'd also like to thank CanVECTOR for organizing this event. And yeah, I am
Thanks, Henry.
take some questions.
Good. Can you flip off your slides, please?
There we
That's
go.
great. Any questions? Oh, we have a question. I can't see
Yeah.
what that is. Oh, it's Jess. Go ahead, Jess.
Hi, Henry. That was a great presentation.
Thank you.
I was just wondering that, like you were talking about tissue factor and factor 7A being a cofactor. And have you looked at factor 7A in terms of your clot lysis assay or any other assays, if they're also having an effect on the results you're looking for?
So just adding 7A to the...
Yeah, the factor 7A.
we haven't looked at that directly in that we haven't just added factor 7A to, no, we haven't looked at that directly yet.
Okay, thank
That's
you.
right.
I'll ask a question, Henry. I think I've probably asked this before, but when the virus acquires the tissue factor, I mean, tissue factor just doesn't travel alone. So are there other proteins that are acquired at the same time?
None that we have looked for directly, but that is that is an interest of the lab. There are some others that, you know, dampen the host immune response. There are a few that, other coagulation proteins that are transmembrane that we would be interested in looking at, but we haven't looked at any of those at this point yet. We've just been focusing on tissue factor and the enhancement of the detection to try to wrap up that whole aspect.
Sorry. Yeah, very good. Okay. Any other questions? It's really interesting. All right. We can move on. Thanks, Henry. To the next speaker. You're going to put that up, Kevin? Oh, good. So Ghanem Juneja is at McMaster University, supervised by Paul Kim, and he's going to speak about temporal changes in biomarkers of coagulation and in COVID. All right, how's that?
Thank you.
I don't want to take up half your time. Okay, go ahead.
No, you're good. 1 second, I'm just going to share my screen. Can you see it?
Yep, that looks great.
Okay, all right. Okay. All right. Good afternoon, everyone. My name is Ghanem Juneja, and I am a second-year master's student in Dr. Paul Kim's lab at McMaster University. And today I will be discussing my research on how temporal changes in biomarkers of coagulation, endothelial dysfunction, and fibrinolysis are associated with illness severity in patients with COVID-19. So just a quick background. So we're all familiar with the severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2, which is the virus responsible for the COVID-19 pandemic. This virus first originated in Wuhan, China, and since its first reports in December 2019, it has spread over 220 countries, infected over 519 million people, and killed 6.26 million people as of today. So as you can see in this image, the SARS-CoV-2 virus directly enters the alveolar epithelial cells through the angiotensin converting enzyme 2 receptors, or the ACE2 receptors. These infected epithelial cells generate a dysregulated inflammatory response due to viral exposure, and it results in a cytokine storm, which leads to endothelial damage. The endothelial damage taken together with the inflammation leads to the activation of the clotting cascade, as well as the dysfunction of the fibrinolytic system. And this imbalance leads to clotting within the lungs that leads to hypoxemia and lung failure, as well as subsequent downstream multi-organ failure. So given these systems that are in play in COVID-19 patients, we were interested in identifying potential biomarkers of coagulation, fibrinolysis, and endothelial dysfunction in order to predict clinical course and outcome in COVID-19 patients. We were interested to know why some of these patients have worse outcomes compared to the others with the disease, and if there are certain biomarkers that do predict a worse outcome, maybe we can intervene earlier on and potentially improve this. So in order to do this, we chose 10 biomarkers that we decided to measure using an antigen assay or a functional assay. So we use an antigen assay to measure the levels of fibrinogen, D-dimer, TAT, soluble thrombomodulin, plasminogen, Pi1, alpha-2 antiplasmin, and TAFI. And then we use functional assays to measure the levels of TAFI-A as well as clot lysis times. So today specifically, I will be talking about the CanCOV cohort, which is the Canadian COVID-19 prospective cohort. And we have 99 longitudinal samples for this cohort. And these samples were collected in the first, second, and third wave of the pandemic. So this cohort is further then divided into 3 different groups. So first we have the outpatient group, which has patients that tested positive for COVID-19, but were required to stay at home and isolate. And there were 23 patients in this group. Then there were the ward patients, which were the patients who were sick enough to require admission to the acute hospital care. And there were 31 patients in this group. And lastly was the ICU patient group, which were the patients that were critically ill and were required were required to admit themselves into the ICU or mechanical ventilation. And there were 45 patients in this group. So the 45 ICU patients, out of those, 24 patients died, which gives it a 53% mortality rate. And in the other 2 groups that I mentioned, there were no deaths. So in order to analyze the findings for this cohort, we took the biomarker values that we measured and log transformed them, and linear mixed effect models were used to compare the trajectories of the three groups. And specifically, we compared the ICU and ward patient groups to the outpatient group from the date of symptom onset. So here you can see that the blue is the ward group and the yellow is the ICU group, and the baseline over here is the outpatient group. Keep in mind that these are projected trajectories because not all ICU patients survive till the end. So in figure, modulin levels were significantly elevated for both the ward and ICU group when compared to the outpatient group. Pi1 levels were significantly elevated, but only in the ICU groups from day 1 and 4, between days 1 and 4. But only in the ICU group from day 25 onwards, and TAFI-A levels were increased over time only in the ICU cohort, with the levels being significant from day 35 onwards. And lastly, fibrinogen displayed similar trends as plasminogen, where the ICU group had significantly decreased levels from day 25 onwards. And the other biomarkers were not significantly different when compared to the COVID-19 outpatient group. So to summarize, the D-dimer and soluble thrombomodulin showed the strongest association with moderate and severe COVID-19 disease state when compared to mild disease. Additionally, Pi1, plasminogen, TAFI-A, and fibrinogen may also be useful in identifying patients who become critically ill. And this cohort, taken together with the other cohorts and samples that I have, has the potential to make a direct impact in COVID-19 prognostication by identifying patients that are at greater risk of decompensation. And lastly, I would like to thank and acknowledge everyone that was involved in the research, particularly my supervisor, Dr. Paul Kim, and our collaborators at TARI at McMaster University and University of Toronto, and the COVID Beacons investigators, and also CanVECTOR for providing me with this opportunity.
That was great, Kenny. Questions? I'm looking for, Ed is jumping in there. Prysdale, go for it.
Somebody else want to go first?
I don't see people.
Go ahead, Ed. I'll ask my
Allison.
question because I bugged Gamine enough, so you can.
Okay, that soluble thrombomodulin thing, I don't think that's ever been reported. Some of those other parameters have been. What do you make of that?
I feel like it is very interesting, and I feel maybe that's why we're trying to validate it using our other cohorts to see if it's actually true, or it's just this specific cohort, and for some reason it was like elevated.
Ganeem, do you think that's maybe telling you something about an endotheliopathy going on in COVID? Do you think that might be unique in COVID? Maybe reporting on something that we might not see in something like flu?
It's possible. It's very possible, yes.
Yeah, that'd be really cool. Maybe something to poke your nose into the literature about.
Yeah.
Yeah, that's cool. Really interesting.
Right.
Thank you.
Elevated thrombomodulin is quite widely reported in COVID-19.
Oh, is it? I didn't know that.
Yeah.
Oh,
yeah.
that's really
We've shown that too with Ganeem's paper as well.
I didn't know that.
Yeah,
Cool.
there's
Very
also some literature out there in general sepsis of using thrombomodulin inhibitors. And
oh, yes, of course, in sepsis, of course. Yeah, I'd forgotten that. Yeah.
I mean, one of the things that we know is the sickest of the ICU patients, there's a proportion of them that went on extracorporeal life support. I don't remember if any on your cohort we did. I certainly know some of the patients we had here in Hamilton did. I wonder if you controlled for those patients that were put on ECMO, because ECMO in itself will consume fibrinogen particularly. So it does, the filter consumes some of those factors. So I'm not sure if you've controlled for that in your data.
So we don't have the clinical data available for this cohort yet, but when we do, I'm sure like we'll sit down and control for all those things, but we don't have the data yet.
Okay.
Patricia?
Great talk, Ganeem. I was just wondering if you, or maybe others in your lab, have you thought about starting to do some functional assays with the samples?
Functional assays, we are doing the clot lysis times and TAFI-A. What other ones were you talking about, sorry?
Yeah, those are the main ones I was thinking of as well.
Yeah, we've done clot lysis assays on these samples and TAFE levels.
Thank you.
Paul?
Yeah, sorry, I just want to add to Allison's comment about ECMO patients. It's absolutely true. I think we have, there's quite a few of the ECMO patients, especially in those ICU died percentage, and it is favoring that. However, we don't have the detailed data to go with it, like the clinical data. In terms of the fibrinogen levels going down, I think so we're showing the trend relative to outpatients, but the absolute face value fibrinogen levels still are elevated. So yeah, they're coming down, and I think they're being, ECMO kind of consuming and just sort of crashing. It certainly is a factor, but they are elevated at face value as well.
Great. Okay, thanks, Paul. Patricia, did you still have a question, or your hands just?
No, I'm
stuck?
fine. Thank you.
Okay. All right. Thank you. I think we should move on. Thank you very much. I just want to remind everybody to make sure you do your evaluations. All right. The link is in the chat box if you've lost it. Okay. Next speaker is Erblin Kenny from McMaster University, supervised by Patricia Liao. And it's immunothrombosis biomarkers for distinguishing coronavirus patients from non-coronavirus patients. All right, so go for it.
Okay, let me just share my screen.
That would be good.
Okay, everyone can see it, right?
Yep, but you need presenter view. Oh, we lost you.
Oh yeah, wait, hang on, no, it's not working for me either. One sec, it disappeared from my screen when I did it.
Okay, not a good sign.
There we go, that should be better, and I'll just switch to interview. There we go, does that work?
Yep, good.
Okay, perfect. Okay, so hi everyone, my name is Erblin. I'm a student in Dr. Patricia Liao's lab, and today I will be presenting my work, which is on immunothrombosis biomarkers for distinguishing COVID patients from non-COVID septic pneumonia patients and for predicting ICU mortality. So severe COVID infection has been linked to a wide variety of cardiovascular disorders, and these include myocarditis, heart failures, dysrhythmias, and thromboembolic disease. And these all arise as a result of multiple mechanisms. So first and foremost, those with pre-existing cardiovascular diseases are at an increased risk of experiencing adverse events after an infection. So related comorbidities are definitely a huge driver of this. Next, the presence of ACE2 receptors on endothelial cells means that there's a significant risk of injury to the vascular endothelium. So that'll lead to its dysfunction, inflammation, and subsequent clot formation. And studies have also found that NETosis can occur in severe COVID infections. And what that means is that neutrophil activation can lead to a process called NETosis, which releases neutrophil extracellular traps, or nets for short. And these are large web-like structures that are made up of proteins and decondensed chromatin. And they're very useful in capturing pathogens by engulfing them and secreting antimicrobials. But the downside is that they tend to promote thrombosis, which is what we're seeing in COVID. And then on top of this, there are also many other biomarkers that have altered concentrations as a result of COVID. So generally, we can see that there are many different causes of the thrombosis that can lead to mortality in COVID patients. So when I started my project back in July of 2020, there were very few studies that investigated the longitudinal changes in immunothrombosis biomarkers, especially in severely ill COVID patients. At the time, most of the papers that were out only focused on 1 or 2 time points. So the objective of our study was to track the trends in a relatively large set of biomarkers in COVID ICU patients over time and compare them to other septic ICU patients that didn't have COVID. And this allowed us to determine the utility of these biomarkers in distinguishing between the COVID and the non-COVID patients. And on top of that, also evaluate the utility of the biomarkers for predicting ICU mortality in these patients. We were able to publish the results of this hypothesis generating study in Critical Care Explorations in December of last year, which is what I'll go over with you in the coming slides. So our initial project involved a relatively small set of patients because, as I mentioned, it was a hypothesis generating study. So 9 biomarkers were measured from plasma samples taken from 14 COVID patients and 19 septic pneumonia patients and 14 agent sex matched healthy controls. The biomarkers that were measured were thrombin antithrombin, or TAT for short, antithrombin, protein C, soluble thrombomodulin, soluble EPCR, D-dimer, fibrinogen, cell-free DNA, or cfDNA for short, and citrullineated histones, or H3-CYT. We collected plasma over roughly 14 days, and we tried to get about 5 time points between those 14 days. But obviously there were some variations due to weekends, holidays, or patients unfortunately passing away. And that accounted for half of our COVID patients and 32% of our septic pneumonia patients. So the first thing we did was graph the biomarker trends over time. For blood coagulation, both COVID and septic pneumonia patients showed increases in thrombin, antithrombin levels compared to healthy controls. For the anticoagulant biomarkers, COVID patients showed lower than normal antithrombin levels, and both groups showed lower protein C levels at first, with sepsis patients returning back to normal while COVID remained low. For endothelial injury, both patient groups showed increased levels of soluble thrombomodulin, while COVID patients showed lower than normal levels of soluble EPCR. For fibrinolysis, both patient groups showed increased D-dimer levels, and COVID patients showed increased fibrinogen levels, while sepsis patients showed lower than normal fibrinogen levels. And then finally, for netosis, both patient groups showed increased cell-free DNA levels, but relatively normal H3-CYT levels, except for a couple of peaks in both groups. And then after establishing those trends in biomarker levels, we then statistically analyzed those results. And on the left, we used receiver operating characteristic analysis in order to generate curves. Calculating the area under the curve told us the extent to which that biomarker was capable of distinguishing between the two patient groups. So in order to be able to definitively state whether the biomarkers were useful or not in distinguishing between the groups, our threshold was that the lower limit of the 95% confidence interval had to have been above 0.5, because that means it's better than random distribution. So in order to distinguish between the COVID-19 and healthy control groups, we found that all the biomarkers except H3-CYT and antithrombin had fairly high area under the curves, or AUCs, and could be used. For COVID-19 and septic pneumonia groups, fibrinogen, soluble EPCR, antithrombin, and cell-free DNA were the most accurate in distinguishing between them. We also checked which biomarkers had utility in predicting ICU mortality, and that was in the COVID-19 and the septic pneumonia groups. And for that, we used another model called a binomial logit model. For the COVID patients, soluble thrombomodulin and H3-CYT were the best 2 biomarkers for doing so, especially when used in combination. And for the sepsis patients, protein C and cell-free DNA were the most accurate biomarkers, once again, doing best when used together. After publishing those results in the previous slides in Critical Care Explorations in December, we started to work on expanding the study. So we're currently validating the results of our hypothesis generating study using plasma from over 100 COVID patients from multiple centers around Ontario and Quebec. And here's just a table summarizing the patients that we have in our validation study. So like I mentioned earlier, they're coming from many different centers, and all of them were severely ill with COVID-19 and were placed in the ICU, where all except the disconnect 1 patients at the bottom were placed on mechanical ventilation. Generally, the disconnect 1 patients are a bit different from the other patients, because whereas the other patients are part of an observational study, the disconnect patients are part of a phase 1 clinical trial where they're actually being treated with inhaled DNase-1. And here I just have a summary of the biomarkers that we've been analyzing for each patient group, and I've marked the completed ones in green. So as you can see, we've made pretty good progress and are fairly close to being done, with only a couple of biomarkers being left to do for some of the groups. One thing to note is that activated protein C is only measured in the Hamilton General Hospital samples and the Queen's University samples. And this is because that assay just requires a separate anticoagulant for the blood when it's initially drawn from the patients. And that's only available in those 2 cohorts. And finally, although we're still working on those biomarker assays for the validation study, we're still able to draw some information from the results we have so far with the help of Dr. Paul Kim's lab, who ran the TAT and thrombomodulin assays on this page. So in our Hamilton General Hospital patients, you can see that there are elevated thrombin antithrombin levels in the graph on the top left, which is a marker of ongoing coagulation in the patient. And this means that we would also be expecting increased levels of activated protein C in the graph on the top right, but we're not really seeing that in most patients. And only in a couple of patients we're seeing those increased levels that we're expecting. And this might be because of the elevated soluble thrombomodulin levels on the bottom left graph. So thrombomodulin is an endothelial protein that plays a role in activating protein C. And since it's an endothelial protein, measuring the soluble levels of it, especially elevated soluble levels of it, means that it's in the bloodstream instead of bound to the endothelium, which indicates that there's some sort of endothelial injury or disorder going on. So that could be one potential reason why we aren't seeing similarly increased levels of APC. And then quickly, just to summarize everything, we were able to find a number of biomarkers that had utility in distinguishing COVID patients from septic pneumonia patients and healthy controls, and also predict ICU mortality. And together, these indicate that there is a difference in the pathophysiology of immunothrombosis biomarkers between COVID patients and septic pneumonia patients. But since our initial results were based on a relatively small sample size, we're currently working on validating those results in a much larger group of patients. So I just want to thank everyone in my lab and Dr. Paul Kim's lab for their help so far in my studies, as well as the researchers from Dr. Douglas Fraser's lab at Western University and everyone at the COVID Beacons team and CanVECTOR for your support. And now I'm ready for any questions you guys might have.
Great, thank you. Can you flip off this? Oh, excellent, excellent. I'm going to start. I have a quick technical question. Cell-free DNA and H3CIT.
Yeah.
Why is there a discordance? I'm not in this field, so why is there a discordance in those markers?
So cell-free DNA generally can be a marker of cell death in general. So, you know, apoptosis, or maybe not apoptosis, but just any sort of dysregulated cell death. But H3CIT is more specific to netosis, which will tell us whether there's neutrophil extracellular traps being formed, which is what we're looking at.
So why do you measure cell-free DNA?
Cell-free DNA, I think it's just better to have a general marker of cell death. It gives us a good idea of how severe the patients are suffering from COVID.
Okay. Alex, thank you. Alex?
I think that was a great presentation. One question I had was whether there was data provided on the existing conditions or like pre-existing conditions or comorbidities that the patients may have had.
Yeah, that's a good question. So because we have so many groups involved in this study, there's basically different answers for your question depending on what groups we're looking at. Our initial group of patients had a fairly good set of data in terms of, you know, comorbidities. I mean, it wasn't fully robust. It's not like we know everything about their, you know, health history, but we were able to see, you know, their clinical characteristics, how severe, whether they were, you know, intubated, whether they were on antibiotics, and so on.
Other questions? I have another question.
Mm-hmm.
In your expanded studies that you're going to doing whatever, are you restricting yourself to alpha variants, delta variants, Omicron? The impression, I shouldn't say that there's not a lot of data, the impression that Omicron doesn't cause a severe disease. Of course, a lot of vaccinated people there, but so how are you managing all that?
Yeah, so these were collected quite a few months ago. So while I don't have information right now as to their variants, and I don't even know if the hospitals collected that information at the time, I think it's safe to assume that most of these patients are either suffering from the initial strain of COVID or the Delta variant, because those were the most prevalent at the time. But I have no information right now as to what exactly each patient was suffering from.
Okay, good. Okay, great. Everyone, thank you very much. That was fantastic.
Thank you.
All right, we'll move on to our next speaker. This is Felix Hong from UBC. The supervisor is Hugh Kim, investigating the role of filament A in platelet shape change. Felix, you're on.
Here.
You should be able to share.
Yep, let me share the screen. All right, can everybody see my screen?
Yep, just have to put it into.
All right, yeah, okay. Good. All right. So thanks for the introduction. So today I'm going to be talking about investigating the role of filament A in platelet shape change. So platelets are anucleate blood cells derived from the cytoplasm of megakaryocytes, and their primary function is to stop us from bleeding upon vessel injuries by forming blood clots. And this activation of platelets is mediated by many signaling pathways, also by many agonists such as thrombin and ADP. Usually in normal physiological conditions, this activation of platelet is tightly regulated. So unfortunately, upon abnormal activation of platelets, it can lead to thrombotic diseases such as arterial thrombosis, which is the underlying causes of heart attacks and strokes, as well as venous thrombosis, which is the common cause of pulmonary embolism. All of these conditions are fatal, so learning more about how platelets are activated will provide us with a deeper insight into the nature of these pathophysiological conditions. So for the sake of my project, I'll mainly talk about platelet shape change. And platelet shape change is one of the earliest physiological response upon platelet activation. And it's driven by a dynamic remodeling of the actin cytoskeleton. And then is followed by platelet granule secretion and platelet aggregation, which ultimately forming a stable blood clot to prevent us from bleeding. And this platelet shape change is regulated by actin myosin contraction. Myosin, as everybody might know, is a molecular motor that burns ATP as a fuel. And when myosin is activated, it will bind to the actin cytoskeleton and causes this contraction, which leads to changes in cell shape. And this activation of myosin is regulated by myosin light chain, or MLC. And when this MLC is phosphorylated, myosin is activated and it will bind to the actin cytoskeleton and causes the contraction. So essentially, when platelets are activated, it will lead to phosphorylation of MLC. And this process is mediated by two very important enzymes. And the first one is called MLC kinase, which is shown in pink here. And the other protein is called, or enzyme is called the MLC phosphatase, which is shown in green. So what happened is upon an agonist activation, for example, by thrombin, it will lead to the activation of MLC kinase, which will then put a phosphate on the MLC, causing the contraction. On the other pathway, which mediated by the Rho A and Rho kinase, their job is to inhibit MLC phosphatase. So when phosphatase is inhibited, nothing is removing the phosphate from the MLC. So together, both of these pathways synergistically allowing for the accumulation of phospho-MLC, leading to actin-myosin contraction, allowing for platelet shape change to occur. And recently, there has been a protein, an actin-binding protein called filament A, has been shown to interact or regulate Rho A function, which lead us to question, does filament A play a role in MLC phosphorylation? More specifically, we hypothesize that filament A could be acting as a major scaffold to recruit all of the necessary components to activate Rho A, which then inhibits MLC phosphatase. This will allow for the accumulation of phospho-MLC, allowing for platelet shape change. And in order to study this, there are actually two major aims for my project. So the first one is to actually find a new cell model to study this MLC and filament A interaction, because this interaction is pretty much conserved in many different cell types. And then the second aim is to basically study this in platelets. But today I'll just mainly talk about the first aim. So aim one is to find a new cell model. So after some digging, I decided to use HEC293 cells to study this interaction because they're relatively cheap and easy to maintain. They're pretty easy to transfect. We can easily make a knockout or a knockdown of a gene. Most importantly, they express PAR1 receptor as shown by this RT-qPCR data. PAR1 is the receptor for thrombin. And also these HEC293 cells seems to be responding to thrombin treatment as shown by this elevated calcium level on the right here. So after that, we obtain some HEC293 cell lines with a stable knockdown of filament A as verified by this Western blot here. So these HEC293 cells do not have filament A proteins being expressed compared to the wild type. So next, after we obtain these cells and verified these are the cells that we wanted to see what happens when we treat these HEC293 cells with thrombin. And what I found was that upon thrombin treatment, there seems to be a higher F-actin polymerization in the wild type, as indicated by the higher fluorescent staining by Alexa 488. And however, in the filament A knockdown cells, as you can see, there are no significant difference in fluorescent staining, indicating that in the absence of filament A, thrombin signaling in these HEC293 cells are somewhat blunted. Next, we want to see if thrombin can induce phosphorylation in MLC. So I've done some Western blot on this. In the wild type, upon thrombin treatment, as you can see, there's an increase in phospho MLC. Again, in the filament A knockdown cells, even with a large dose of thrombin, there is essentially no difference in the non-treated group. And this result is comparable to the control without filament A knockdown. So further proving that when filament A is knocked down, MLC phosphorylation and thrombin signaling is inhibited, despite having more MLC in the basal level. So aim 2 is to actually study this in platelets. So in order to study this, our lab has generated a platelet-specific filament A knockdown in mice by using a tissue-specific prelox system in the megakaryocyte lineage. So these mice, the experimental mice that we have no filament A inside of their platelets. And currently the experiments are in progress, and hopefully in the next few weeks, I'll be getting more data on this. So to sum it all up, so what I found so far was that when thrombin-treated HEC293 cells induce more F-actin polymerization in the wild type compared to the filament A knockdown cells, and MLC signaling is somewhat weakened in the filament A knockdown HEC293 cells. But then again, the mechanism of how filament A regulate MLC phosphorylation has still remained to be proven. And also, the experiment with the filament A null mouse platelets are currently under progress, and probably going to get more data in the next few weeks. And with that, I would like to thank all of my lab member for being supported in what I do, and thank you for all of the funding agency and CanVECTOR for hosting this event. And with that, I would like to thank everyone for listening, and I'm happy to take any questions. Now,
Thanks, Felix. That's great. Any questions?
Stop sharing.
Here we go. Any questions?
Hi, great talk. I have a quick question. So,
Valerie, yeah.
Do you think that filament A will play a role in platelet-generated contractile forces?
I would believe so, because filament A is pretty much binding to the, normally the regular role of filament A is to bind to the active cytoskeleton, and it kind of stabilizes the whole network. So I would assume that it will pretty much causes contractile forces in the platelet as well.
Great, thanks.
Any other questions? Okay, so then we'll move on to the next speaker, which is, here we go, Jaskirat Arora of McMaster University. The supervisor is Alison Fox-Robichaud, and this is an observational pilot study investigating the role of coagulation in the diagnosis of sepsis in the emergency department.
Thank you, Ed.
Pleasure. Okay.
I'll start sharing my screen. Okay. Here you go. Can everybody see your screen?
Excellent. Yep.
Perfect. So good afternoon, everyone. My name is Jaskirat Arora. Today, I'm going to be talking about my project, Sepsis CD, which is investigating the role of coagulation in the diagnosis of sepsis in the emergency department. Before that, a little bit about me. I'm a PhD candidate at McMaster University, and I'm currently working under the supervision of Dr. Alison Fox-Robichaud. So sepsis is an inflammatory condition that progresses from a state of infection during time zero. We need biomarkers to diagnose this switch from infection to sepsis, and we believe it is linked between the inflammation and the coagulation pathways. These two pathways are linked by the viable antibodies. So we hypothesized that the coagulation biomarkers can improve the difficult diagnosis of sepsis in the emergency department. So upon endothelial injury or activation in sepsis, viable polarity bodies are secreted that release P-selectin and VWF multimers. P-selectin mediates neutrophil recruitment by facilitating neutrophil rolling on the endothelium. Activated neutrophils release extracellular traps that consist of cell-free DNA and histones. Netosis can act as a protective mechanism by entrapping the microbes in its mesh-like network to eliminate them. However, if there is excess netosis, it can cause coagulation dysfunction by downregulating the natural anticoagulant of the body, protein C, which leads to a state of thrombosis. Viable polarity bodies also release VWF multimers that are important in platelet aggregation and thrombus formation. ADAMTS13 is a protease that acts as a scissors to cleave the VWF multimers into smaller fragments and control platelet aggregation. However, in sepsis due to ADAMTS13 deficiency, the uncleaved VWF multimers can form aggregates with platelets, resulting in thrombus formation in the capillaries. So we believe that VWF multimers and netosis can predispose the body to VTE and are essential in the early diagnosis of sepsis. So our study, Sepsis ED, is an observational cohort study that will recruit 250 adult patients with sepsis from the emergency department of 2 hospitals, Jurwinski and Hamilton General Hospital. Samples are collected at 2 time points. Time point 1 is usually before the antibiotics are given, and time point 2 is 4 hours after the initial sample collection. So the aim of our study is twofold. One is to investigate the diagnostic potential of coagulation markers such as cell-free DNA, protein C, histones, ADAMTS13, and VWF levels. 2, to create a panel of biomarkers that includes the standard biochemical test, patient clinical scores such as their vital signs and organ dysfunction score, together with the lab biomarker analysis. So far, we have gathered data on 230 patients, including the left panel of 100 septic patients having 45 females, aged 74 years, and the right panel represents 130 non-septic patients with 64 males aged 71 years. Among the septic cohort, cultures were drawn in all patients except 1 patient, and among these, positive cultures were observed among the 58 patients. And antibiotics were also given to majority of the septic patients. In the non-septic cohort on the right side, cultures were drawn in 116 patients among the 130 non-septic patients, with positive cultures observed in 69 patients. And again, as you can see, that antibiotics were given in about 80% of the patients. So now I will be showing you Violin plots on biomarker analysis. A little bit about the Violin plots is that the Violin plots show median and the interquartile range, and with the tail end showing the higher and the lower range of the biomarker levels. So figure 1 shows the cell-free DNA levels at 2 time points. The patients in dark and light red are the septic patients at time point 1 and time point 2. Similarly, the patients in dark and light blue are the non-septic patients at time point 1 and time point 2. So a previous ICU study, Dynamics, that was recruiting septic patients from the ICU, saw higher cell-free DNA levels in the septic patients. But in our cohort of the emergency department patients, we do not see this change. We also looked at the protein C levels at 2 time points. The protein C levels were not lower in the septic patients compared to the non-septic patients. However, these levels declined in the septic patients 4 hours after admission to the emergency department. As you can see, a difference in the Violin plots in the 2 red. Now looking at the marker ADAMTS13 and VWF. So ADAMTS13 levels decreased considerably in the septic patients compared to the non-septic patients at both the time points, which is seen as a comparison between the red and blues. And these levels were also lower than the healthy individuals, which is seen by the dotted lines. However, the ADAMTS13 levels in the septic patients do not decrease 4 hours after admission to the emergency department, which is in contrast to what we saw for the protein C levels. So lower ADAMTS13 levels would have meant that there are more VWF levels as there is less ADAMTS13 to cleave it. The VWF levels were higher than the healthy controls. However, their levels were not significantly different between the septic and the non-septic patients. So overall, we conclude that ADAMTS13 levels decrease in the septic patients compared to the non-septic emergency department patients and to the healthy individuals. This emphasizes the importance of immunothrombosis in early sepsis. Therefore, the biomarkers of coagulation response could be important in the diagnosis of sepsis. I would like to thank my supervisor and the entire sepsis ED team and my lab members and the Liao lab members. Thank you, everyone. Happy to take any questions or suggestions.
Thanks, Jones. Questions? Oh, we have a question.
Hi, Erbelin.
Yeah,
Hey.
go.
I'm just wondering, have you looked at how ADAMTS13 activity might impact your results or your findings?
So that's a very good question. We have discussed about it a lot. Because the antigen levels of ADAMTS13 tell us the amount of ADAMTS13 that are present in the blood, but it doesn't tell if those levels are active or not. But then Colin Kretz lab members did a study of using ICU patient samples, and they saw that the ADAMTS13 levels and the activity don't have a correlation between them. So that is something we can definitely explore in our study, what happens in the emergency department patients.
Thank you.
So to go on, follow up on that question, because I was going to ask why are the levels low in sepsis? Are there any mechanisms described? But you're saying that the antigen levels are not reduced, but the activity levels are reduced. Is that right?
So we have only looked at the antigen levels, and we see that the antigen levels are reduced. We have not looked at the activity levels in the emergency department patients.
Okay.
So the mechanism that it's actually my aim3, where I actually propose that the ADAMTS13 levels are reduced because of their interaction with the components of NETs, maybe because of their oxidative action.
Any other questions, comments? Okay, if not, we will move on.
Thank you.
To our penultimate speaker, actually. So the next student speaker or trainee speaker is Lisa Yamura from the University of Calgary. Supervisor is Dr. Prism Schneider, and topic is hypercoagulability in patients with metastatic bone disease as determined by TEG, a prospective study. All right, Lisa.
Great. Thanks, Dr. Conway. Can you guys see my presentation?
Just not in presentation mode.
Here we go.
That's good.
So, So what exactly is metastatic bone disease? Well, metastatic bone disease occurs when patients have cancer that then progresses and it spreads towards the bone. And some cancers, for example, breast, prostate, and lung cancer commonly do this. And then patients develop metastatic bone disease. So you can see in this X-ray image here, a patient with metastatic bone disease, and it's caused a lot of destruction to the bone and affected the quality. So oftentimes this necessitates orthopedic surgery, as shown here, this patient in the postoperative X-ray here. So unfortunately for patients with metastatic bone disease, both malignancy and orthopedic surgery are independent risk factors for venous thromboembolism, or VTE. And VTE encompasses two clotting complications: deep vein thrombosis, or DVT, which is development of blood clots in the legs, which you can see in this Doppler ultrasound image. And it also encompasses pulmonary embolism, which occurs when blood clots migrate to the lungs and cause blockages in the pulmonary arteries. So we know that patients with undergoing orthopedic surgery who also have metastatic bone disease are at very high risk of VTE complications. So orthopedic surgeons will prescribe thromboprophylaxis after surgery, and that's medication to prevent incidence of VTE. However, because we don't know the extent and duration of hypercoagulability in patients with metastatic bone disease, this has led to, unfortunately, a lack of clearly defined guidelines for thromboprophylaxis for these patients, despite the high risk. So that leads us to our aim at our research group, where we were really interested in learning about the extent and duration of postoperative hypercoagulability in patients with metastatic bone disease to fill this knowledge gap. And we also want to be able to use a thrombelastography or TEG analysis to have values in which we can compare patients who have VTE and those who do not in patients who have metastatic bone disease and require orthopedic surgery. So for a thrombelastography or TEG, what it is it's a tool which measures the clotting process, and it's a cartridge-based test. So we take a small sample of the patient's blood, put it into the cartridge, and insert the cartridge into this TEG machine shown here on the left. And as the machine runs the test, the blood transitions from the liquid state over time into a gel-like state during the clotting process. And the TEG machine measures maximal amplitude. It's measured in millimeters. Maximal amplitude, which is shown here, is an indicator of clot strength. So when the TEG tracing changes shape based on coagulation status. So in this example, in the red tracing, this would be a patient who is hypercoagulable. So you can see that the maximal amplitude here is higher, and that is associated with a stronger clot that's been formed and increased VTE risk. And previous published research has shown that in orthopedic trauma patients, a maximal amplitude of 65 millimeters or greater is indicative of hypercoagulability and associated with a nearly 4-fold increased risk for VTE. So for our prospective cohort study, we enrolled adult patients who presented to Foothills Medical Center requiring orthopedic surgery to treat pathologic fracture due to metastatic bone disease or hematological malignancy of bone, which includes multiple myeloma. And we excluded anyone who had primary bone tumors, anyone who is currently pregnant, and anyone who is undergoing treatment for VTE already. And just important to note that incidence of VTE was a study endpoint for patients who did enroll and participate in our research. So for our study, we collected blood samples at 8 time points. Our first sample was taken preoperatively. We followed patients during their hospital stay on postoperative day 1, 3, and 5 by collecting blood samples on those days. And we also saw patients when they came back to Foothills for follow-up visits with their surgeon, 2 weeks, 6 weeks, 12 weeks, and 24 weeks postoperatively. And because patients are so high risk for VTE, we also paired our postoperative day 3 TEG analysis with an ultrasound. When patients came back for follow-up visits, we also conducted patient interviews, and we asked patients about adherence to thromboprophylaxis, and we also asked them about incidence of VTE after they had been discharged. So for our study, we've enrolled 28 patients who've completed the study so far. Our mean age is 68, and about half of our patients were female. The most common primary cancers amongst our patients were breast, colorectal, and lung cancers. And here we have our maximal amplitude graphed across our study time points. We had 4 patients experience VTE, all happened within the first week postoperatively. And just as a reminder to recall that VTE incidence was a study endpoint. So we see our patients who had VTE in the red line. Our patients who did not have VTE is shown in the green line. And our shaded box shows the hypercoagulable state. So we can see that patients who experienced VTE complications were more hypercoagulable than patients who did not have VTE. However, in the patients who did not have VTE in our green line, we can see that starting at postoperative day 3, we see that hypercoagulability, 83% of patients were hypercoagulable here. This peaked 2 weeks postoperatively with nearly 90% of our patients in a hypercoagulable state. And even as far as 6 weeks postoperatively, we can still see that they are hypercoagulable. The mean MA was 65.1 at this point. And this is interesting because typically thromboprophylaxis is provided or prescribed for a 4-week period. So patients would have completed their thromboprophylaxis prescription course already. So we can see that hypercoagulability is prolonged in patients with metastatic bone disease. And so what we've uncovered so far is that with our serial tag analysis, we've been able to identify prolonged hypercoagulability. We've been able to see that patients who have VTE complications tend to be more hypercoagulable than those who do not. And now that we've been able to look into the extent and the duration of hypercoagulability, future investigation into potentially how this is happening, why are patients hypercoagulable, and what's driving this increased VTE risk would be interesting to look into in the future. And I just wanted to thank my supervisor, Dr. Schneider, as well as everybody on the orthopedic trauma service and orthopedic trauma research team, and of course, CanVECTOR for their support to make this research possible. And I thank you so much for listening. And if anyone has any questions, I'm happy to answer them.
Thanks, Lisa. That was fabulous. Flip off your slides, please. Questions? It's really interesting.
I just had a quick question, Lisa. Thank you for the presentation. Regarding the timing of thromboprophylaxis, perhaps, this is not directly related to what you did. However, I'm wondering, is there something in the literature about these patients with MBDs and maybe specific types of cancers, and what timeline would be an optimal point after surgery to, in fact, give these patients thromboprophylaxis? Is there anything out there?
Specifically for metastatic bone disease, it's been difficult. We did do a systematic literature review, a meta-analysis, and we were able to see that there was quite a bit of variation in what's been prescribed. I know typically patients are recommended to start thromboprophylaxis about 12 hours post-operatively. Typically, it ends up being the next day that they would start. And it is typically low molecular weight heparin from our literature review. We found that was commonly favored in terms of prescription. But again, for the duration, it really, really varied across all of the studies that we found. Some patients were not even receiving any thromboprophylaxis. So that's why this is something that we're really, really interested in, because it just seems like there's no real consensus. And a lot of our orthopedic surgery patients are receiving thromboprophylaxis based on fracture type. So patients who experience hip fracture are grouped together, patients who experience femur fractures are grouped together in terms of prescription. And for patients who are higher risk, like patients with cancer or metastatic bone disease, the recommendations just suggest higher risk patients, we should extend this for longer, but no real direction in terms of what that should look like.
Yeah, that's too bad. I mean, yeah, it does. Thank you. It's just too bad that there's no consensus, unfortunately. And I hope there's going to be more studies coming out to sort of look at that and determine that. But yeah, thank you.
Patricia?
Great talk, Lisa. That was very interesting. I just have a general question on the thromboelastography. So there are different types of hypercoagulable states, for example, too much procoagulants or insufficient amounts of anticoagulants. Does the TAG profile give some insights into like the underlying mechanisms of the hypercoagulation?
Yes, so I only presented the results of our global TAG analysis. So we have our cartridge-based TAG technology, and the platelet mapping cartridges are able to look at the platelet receptor pathway contributions. So we're able to look at the adenosine diphosphate pathway and the arachidonic acid pathway. So that can kind of give us a little bit more information in terms of mechanisms as well. And I only presented one value that we're using as our key indicator here for our research, which is maximal amplitude. But the TAG machine, even for the global analysis, which is what I presented, it does give us other values that we haven't had the chance to look into yet, but it can give us information about fibrinogen function and things like that. So there is a lot of information that we can glean, and we're just kind of looking at one specific parameter related to clot strength right now.
Okay, and my second quick follow-up question is, in your patient population, did you have a group that did not have malignancies?
We don't have a control right now, but that's definitely something that we're very interested in having at some point. But right now, we just have our patients with metastatic bone disease in this current study.
Okay, thank you.
Me. Okay, thanks, Lisa. That's great. I think we'll move on. I just want to remind everybody to do your evaluations at that link which is somewhere in the chat as well. All right. And we're going to move on to our keynote presentation. This is Dr. Valerie Tutwiler is an assistant professor at Rutgers University in the Department of Biomedical Engineering. She received her BS and Master of Science in Biomedical Engineering from Drexel University, and then conducted research in developing a microfluidic model of heparin-induced thrombocytopenia at Children's Hospital of Philadelphia, a leading center for that disorder. Dr. Tutwiler then conducted her PhD studies between Drexel and the University of Pennsylvania on the kinetics and mechanics of contracting blood clots, again with mavens in the field. She completed her PhD in 2017 and has conducted research at the University of Pennsylvania, where she studies the structure and mechanics of blood clots. Her work is being funded by the NIH, the American Heart, and the American Society of Hematology. And outside the lab, she enjoys playing tennis, reading a good book, and baking. So I don't have a title, so maybe she's going to bake for us. Otherwise, maybe talk about something else.
Yes. Well, thank you for the introduction, and thank you for the opportunity to present our research today. So I will be talking about the structure and the mechanics of blood clots in health and disease. Everybody can see the slides, right?
Yep, perfect.
All right, great. So one of the reasons that, I've been interested in blood clotting throughout my training, and now that I've started my own laboratory, is that it contributes to some of the leading causes of death worldwide. And in particular, the focus of the talk today will be more on the cardiovascular disease side of things, which accounts for over 17 million deaths per year, and is for 1 in every 3 deaths in the US. So as all of you probably know, if you get a cut, right, you want your blood to clot because you want it to stop bleeding. And so this occurs through a process called hemostasis. Now, the hemostatic balance, it sits on a very finely tuned scale effectively, where on one hand, if you have not enough clotting or too much breakdown of the clot, you get bleeding. That can occur in genetic conditions such as hemophilia, hemorrhagic stroke, or following traumatic injury. On the other hand, if you have too much clotting or not enough breakdown of the clot, you can develop thrombotic conditions. So heart attacks, ischemic strokes, venous thromboembolism, for example. So my lab studies both the bleeding and the thrombosis side of this regulation. However, the talk today will focus more on the thrombosis side of things and the mechanisms that regulate some of these processes, specifically from a structural and mechanical standpoint. So it's important that blood clots are structurally, mechanically, and enzymatically stable during their, in order to perform their functions in a case of hemostasis, or to resist embolization in the case of thrombosis. So just to give a quick overview of some of the key players that I'm going to be talking about today during the presentation, again, which many of you or all of you are probably very familiar with. So the components of a blood clot that I'll be talking about are fibrin, which can be seen here in this colorized scanning electron microscopy image, in blue as the polymeric network that holds the clot together. We have red blood cells, which you can see kind of take up a large portion of the volume of the blood clots, and we'll talk about them during the presentation as well. And then we have platelets, which here are colorized in yellow and primarily responsible for a lot of the contraction aspects that I'll talk about during the presentation as well. So blood clot contraction is the volume shrinkage of a blood clot. It's driven by platelet fibrin interactions. It results in the shrinkage of the overall clot volume. So here you can see an uncontracted clot compared to a contracted clot. And so it results in the compression of the red blood cells into the core of the clot and the expulsion of the blood serum outside of the clot. So clot contraction has been implicated to play a role in a variety of different processes, including wound healing, hemostasis and preventing the blood flow or the loss of blood following the formation of a clot, and then in thrombosis, and particularly in understanding the occlusivity of a thrombus. So we have previously shown that in patients who develop thrombosis, ischemic stroke, and venous thromboembolism as examples, we see that when compared to healthy donors, the patients who have thrombosis, in this case VTE, have a reduced extent of clot contraction, meaning that their thrombus is more occlusive into the blood vessel than their comparative healthy donor clots would be. So these experiments are done in the blood from these patients in an in vitro assay. Now, interestingly, we see that in patients who go on to develop a pulmonary embolism, in addition to a deep vein thrombosis, that extent of contraction is reduced even further compared to those patients that have a deep vein thrombosis alone. And so that points to the potential role that clot contraction can play in the development of pulmonary emboli and in the course and the outcome of thrombosis. But it still leaves the door open to answering questions that get at the mechanism of why some of these differences might be seen, how clot contraction could influence pulmonary embolism risk in addition to the occlusivity of the thrombus. And so particularly, we're going to talk about the enzymatic and the mechanical stability and how that's related to clot contraction and to thrombosis outcomes as well. So first we'll talk about the enzymatic stability. So blood clots must be enzymatically stable, right? Unless you want them to be degraded in the case of hemostasis. So the degradation of blood clots occurs through a process called fibrinolysis. And so in the case of internal fibrinolysis, that's what would happen during the normal pathophysiological process of fibrinolysis. So if you have a blood clot that is formed, you would want that clot to then ultimately be able to be degraded when you want it to be degraded, right? If it degrades too early then or too much, then you would have bleeding conditions. And this occurs through the plasmin-mediated cleavage of the fibrin network through related to TPA and plasminogen. In contrast, we have external fibrinolysis. So this is where, from a clinical standpoint, thrombolytics would be administered to a patient where a blood clot is already formed, and then TPA is administered to a patient where the clinicians would want to drive the resolution of the blood clot. So fibrinolytic agents are used to treat thrombotic conditions, but it's known that older clots are more resistant to lysis following treatment with TPA, and that the efficacy of thrombolytic treatment is dependent on both the age and the size of the thrombus. And so we propose that clot contraction, and in particularly the fibrin density, plays a role in that time-dependent efficacy of antifibrinolytic treatments. And so there are a couple of important changes that occur to the structure of blood clots that could tell us, what's happening following clot contraction that can influence the outcome of thrombotic conditions. So clot contraction results in the compression of the clot volume, as I said before, but you have structural changes where you have the movement of platelets and fibrin to the outside of the clot. You can see here a collection of that fibrin, the polymeric fibrin network, and red blood cells that are compressed into the core of the clot and form a tessellated network of red blood cells. So we show that during the contraction process, you get a redistribution of those platelets and red blood cells, right? So you can see here in a scanning electron microscopy image that most of the fibrin is on the exterior or right at the edge of the clot. And the further we go into the clot, the smaller percentage of fibrin that we have. Whereas if you look at the interior of the clot, you can see that the further that you go into the interior of the clot, the more red blood cells, the more area that's covered by red blood cells, and the more tightly packed those red blood cells are, which can be seen by the decrease in the number of pores. So importantly, we wanted to confirm that clot contraction drives the spatial redistribution of the components of the clot. So you can see here that you have a contracted blood clot. Using histology images, you're able to see an outer edge region, and then you're able to see an interior region, clearly two separate regions. If we impair contraction using blebbistatin, for example, which blocks the myosin contractility, you can see that you no longer get those two distinct regions of the clot, but rather you have a homogeneous clot. And importantly, this redistribution occurs not only in vitro clots, but also in thrombi that are extracted from both arterial and venous thrombosis. So again, pointing to the importance in understanding how these structural changes are going to affect the course, the outcome, and the treatment of these clots. So we have previously shown that clot contraction influences the process of external fibrinolysis, right? So where TPA is administered to provoke the resolution of blood clots in thrombi, in patients that have developed thrombosis. So here I'll show you a video that looks at the assay that we use to study the external fibrinolysis process. So in the first video, you're going to see that there's no TPA that's added. You'll see the clot form and contract, but no TPA is being added in that left image. In the right image, you will see that TPA has been added. And so you'll be able to see that it contracts, but then once TPA is added, the clot starts to break down. So we're able to look at the optical tracking of this process and distinguish differences without TPA and with TPA to track the breakdown of the clot. So this can be done either through optical tracking or through the release of radio-labeled fibrinogen. We're able to see that when we impair contraction compared to normally contracted clots, and clots that have contraction impaired have the increased release of fibrin degradation products, telling us that contraction does something to impair external fibrinolysis. But from a mechanistic standpoint, we didn't know what contraction was doing to impair this process. And so that's when we turned back to those structural changes that occur following the process of contraction to be able to further probe what's happening in these particular instances. So as I mentioned before, volume shrinkage of the clot results in the compaction of red blood cells in contraction compared to uncontracted clots. As a reminder, we get the spatial redistribution of fibrin in blue to the edge of the clot, seen here in contracted images where it's more uniformly distributed in clots with impaired contraction. And we get densification of fibrin within this peripheral region in contracted clots, whereas in clots with impaired contraction, the fibrin network is not densified as much. So experimentally, it's hard to isolate these individual changes to be able to probe the mechanistic reason behind this impaired fibrinolysis of contracted clots. And so in order to get at this question, we take a combined experimental and modeling approach to studying fibrinolysis. So in collaboration with Brittany Banish at the University of Central Oklahoma, we have been working on modifying her combined micro and macro scale of fibrinolysis, which was previously focused just on the fibrin network, to be able to include the effect of red blood cells and all of those structural changes that I just talked about on the previous slide. And then we're using turbidity studies to look at changes in the optical density to be able to look at external fibrinolysis and how some of those key changes affect the process of fibrinolysis in contracted clots. So first and foremost, we wanted to develop modeling simulations that that reflected the changes that we were seeing in contracted and uncontracted clots. So in our contracted clots, we take those same changes where we have a volume shrinkage, which compresses the red blood cells. We have a redistribution of the fibrin network to the exterior of the clot and a densification of that fibrin network as well. So we're able to independently vary the amount of fibrin that's in the exterior portion of the clot, the volume of the clot, and the density of that peripheral region. And so we can see that I can show you a video of what this modeling simulation looks like, which looks, you'll see, very similar to what you saw in the previous slides with the experimental breakdown of the clot, where you can see the fibrin fibers go from black, which means that they're present, to green, which means that they're being broken down. So the first thing that we did was to validate our model of fibrinolysis to show that the experimental and the modeling results reflected each other, where we saw that contraction impaired the degradation rate and extended the time to complete lysis, showing that external fibrinolysis is reduced in contracted clots, both in our modeling simulations and then, of course, in the experimental results that I showed you previously, which validates our model and then allows us to move forward and to further probe that mechanistic understanding of what's happening. So we were able to vary the volume shrinkage, the redistribution, and the densification independently in our modeling system in a way that's not easily feasible experimentally. We saw that the redistribution of the fiber network had a very limited effect, if any. The volume shrinkage of the clot had a slight effect. You can see that actually it was a slight effect, but in the opposite way that we would have expected, where the contracted clot, which is this green plot here, contracted slightly faster than the uncontracted clot. And then if we look at the simulations where the fibrin was densified, we see that the denser fibrin network, which is in blue here, compared to a looser fibrin network, resulted in that delayed fibrinolysis, which told us that fibrin densification limits fibrinolysis from mechanistic understanding, and that could be a key player in driving that impaired fibrinolysis in thrombi and in contracted blood clots. We then went back to look at experimentally how fibrin density would alter fibrinolysis. Using turbidity studies, we were able to vary the density of the fibrin network, so the least dense being in blue and the most dense being in purple. And if we look at the time to 50% lysis in this case, we see that increasing the fibrin density or the fibrin concentration results in an increase in the time to 50% lysis. If we compare our modeling simulations with our experimental results, so blue is the experimental results and red is the modeling simulations, we see that both the modeling and the experimental results show similar trends, that fibrin density does in fact, increasing fibrin density does in fact impair fibrinolysis, supporting our mathematical modeling findings, and then of course helping us to probe the mechanism that results in impaired fibrinolysis following clot contraction. So that covers how the enzymatic stability of blood clots could be altered by the process of clot contraction, and the importance of understanding how the structure influences the enzymatic stability. But the second portion of this is that blood clots must be mechanically stable. So for this portion of the talk, we're going to focus more on the fibrin network. So fibrin is a naturally occurring biomaterial that gives the mechanical and the stability, the structural and mechanical stability to blood clots. So again, you can see it here in this kind of teal color, but that's what provides that stability to blood clots. So fibrinogen, which is a blood plasma protein, is the monomer that ultimately comprises that polymeric fibrin network. And the fibrin, when it is polymerized, it forms a structural hierarchy, right, where you have your monomers forming protofibrils and then forming that polymeric fibrin network. And that's how it provides the structural and mechanical stability to the blood clot. So it's important to understand the mechanical properties of blood clots and thrombi. Many of the studies that have been done in the past focus on the viscoelastic mechanical properties of the material. And it is known from these studies that patients with both bleeding and thrombotic conditions have altered mechanical properties, and some of which we heard about in the earlier talk before me. The mechanical properties have the potential to determine whether or not embolization of a thrombi or bleeding will occur. And the response to many treatments are determined by the clot's mechanical properties. So it's important to consider, from both a treatment standpoint, as well as from understanding the propensity for embolization, what the mechanical properties of the clots and the thrombi are. So it's important to note, though, that stiffness does not equal strength. So stiffness, which is what you would measure through some of the viscoelastic mechanical properties, is the degree of the resistance to deformation under an applied load. So if we look at the examples here, rubber, wood, and glass, we'll see that they have increasing stiffness. But strength is the ability of a material to withstand an applied load without breaking or rupturing. And you can see, if we look at the strength of these individual materials, that they are not directly related to the stiffness of these individual materials. And that leads us to the importance of understanding the strength of blood clots, and particularly to understanding the strength of blood clots with respect to embolization. So embolization results in an increase in mortality risk. Right? So if you have a relatively stable thrombus in your leg, but then a portion of it breaks off and travels to your lung, causing pulmonary embolism, that results in a higher increase in mortality risk. So in order to understand the breaking of blood clots and of thrombi, we turn to some classical mechanics techniques and fields, and looking at the rupture, the resistance to rupture, or the fracture mechanics of a blood clot, because that strength is what's going to determine if the blood clot breaks apart or not. So if you think of a thrombus that is protruding into a blood vessel that has some type of a defect or a crack in their surface, as the blood flows across that surface, it's going to apply forces to this edge right here. And that can result in the breaking off of the thrombus, which can then travel downstream and become lodged. So in order to do these mechanical testings of plasma clots, again, we're turning to classical mechanical engineering techniques that have been used on things like rubbers and metals to understand their mechanics. And so this is a plasma blood clot with a crack inserted in the perpendicular direction to loading, and then loading will go in the vertical direction. So I'll play the video, and as I play this video, you'll see the pulling of the blood clot while this force strain curve is being propagated next to it. So you'll see, as we're applying strain to the network, we see relatively little changes in what's happening to this crack, with the exception of it expanding in the vertical direction, up until when we hit a critical point. And then you can see that as soon as we hit that critical point, you get a kind of rapid unfolding of that fibrin network and the breaking of the blood clot, which points to the fact that there's something critical that's happening there that then triggers the rupture of the remaining fibrin network. So toughness is what is the material property that determines the resistance to the rupture. And so we're able to take those same force strain curves and look at what's happening, where we see fiber alignment happening first, then reaching a maximum force, and then ultimately breaking rather rapidly, as you can see here, where once we hit that maximum force, the clot broke very quickly. And that starts to tell us some about the toughness of the material. But first, we had two very important observations. First, the fibrin is mechanically weak in the presence of a defect. So in gray, we have a sample that has no crack, whereas in black, we have samples with different degrees of crack lengths. And you can see that all of the samples with cracks are breaking at much lower forces than the sample without a crack, pointing to the fact that cracks induce weaknesses into the clot. And this is important because the defects occur in blood clots. So from the SEM images, you can see that the clots are not homogeneous. And it's also important to remember that fibrin is enzymatically degraded as well, which we talked about in the first part of the presentation. And so that can start to break up some of the fibrin network as well, forming some of those defects. The second important piece of information that we gained is that toughness is a material property. That means that it doesn't vary based on the shape or the size of the sample or the crack, which tells us that information that we're gaining from these in vitro experiments can then be used to provide insight into in vivo blood clot behavior. So we're able to look at what's happening in these blood clots or in these fibrin network. And we see that fibrin fibers align in the area immediately ahead of the crack tip under strain. So here, using confocal microscopy images, you can see areas removed from the crack tip and immediately ahead of the crack tip. You can see that you get alignment of the fibrin fibers, which is quantified here as an alignment parameter with increasing strain. And you can also see that you get a densification of this fibrin network within this region as well if you look at the mean fluorescence intensity. So we again use both experimental and theoretical modeling to probe the mechanisms and understanding what's happening within this process. So we have collaborations with both Prashant Purohit at the University of Pennsylvania and Valery Barsukov at the University of Massachusetts Lowell to look at the theoretical side of the fibrin network rupture. Here I'm showing simulations from the Purohit lab at UPenn. And we can see that the experimental results and the finite element modeling results line up well, both in terms of visualization and in terms of force displacement curves. So we're able to see the alignment, the fiber alignment ahead of the crack tip, both in the experimental results, which is what I showed you in the previous slide, as well as in our theoretical simulations. But the model's able to take it a step further, and it's able to see that there's molecular unfolding in this region ahead of the crack tip. And importantly, that the strain in this region immediately ahead of the crack tip is similar regardless of the crack length. And so what that tells us is that there's a critical strain at which when the fibrin fibers are placed under it, they break, and then they start to break cooperatively because that strain is being experienced by all of those fibers. And that's why you see that effective unzipping that's happening across the crack tip region once it starts to break. And that tells us a lot about the mechanism of rupture, and that can also be used to go back and then inform what's happening in the body. But we've been looking at how fibrinogen concentration can influence this mechanism of toughness and if it changes or increases the toughness or the resistance to rupture. Because as I'm sure many of you know, the fibrinogen concentration varies across pathological conditions, where it is lower in the case of bleeding and higher in the case of thrombosis. And so importantly, we see that the fibrinogen concentration does in fact influence the toughness of the clot, where with increasing fibrinogen concentration, you have an increased toughness or clots that would be more resistant to rupture. This is important in starting to think about some of the potential mechanisms for embolization, both here, but also if we think back to how this could affect clot contraction. So we've previously shown that fibrinogen concentration, increasing fibrinogen concentration results in a decrease in the extent of contraction. And as I showed earlier in this presentation, a decreased extent of contraction is associated with an increased risk of pulmonary embolism. So it's important to consider kind of the interaction between a lot of these things that are happening, right? Because the thrombosis process is going to be very complex, where the structural components, the mechanical components, and the enzymatic components are all interplaying with each other. And that's going to influence the course and the outcome of thrombotic conditions. So just in conclusion, fibrin must be mechanically and enzymatically stable enough to resist rupture. Clot contraction has the potential to influence embolization because of its destructive nature, its interaction with fibrin density, and its interaction with the propensity to result in fibrinolysis. Increasing fibrin density results in an increased resistance to rupture and an increased resistance to external fibrinolysis. So in conclusion, I would just like to thank everybody in the lab that works on aspects of this project related to blood clot structure, fibrin mechanics, and blood clot resolution. So everybody in my lab, as well as our collaborators at the University of Central Oklahoma and the University of Pennsylvania, and then of course our funding sources as well. And I am happy to take any questions.
Thank you, Valerie. That was fantastic. It's really great to see. It just illustrates how potentially powerful biomedical engineering can interface with real biological questions. Do we have any questions? So while people are thinking, I just had a question. How much do we know about the inhomogeneity of blood clots, say venous thrombi, sitting in the in the leg?
Yeah, so I think that is definitely something that is really important to continue to study. There was a recent paper out of the Weissel lab that looked a bit at how the composition of thrombi differs in venous versus arterial versus pulmonary emboli, and a little bit in terms of the non-homogeneity of those clots. And I think it's important to look at, consider, and to understand, and probably needs much more investigation from both that structural or mechanical stability, as well as the potential to resist fibrinolysis, right?
Yeah.
And so I think that's definitely a great area that needs to be investigated more.
That's why we have young investigators here.
Yes.
Questions?
Valerie, your model is mostly pure fibrin. Do you have the capacity on that engineering model to add cellular material in it to see how that might impact? And the other thing that, when I look at those scanning EM pictures of the fibrin clot, it reminds me of work that Patricia and I have done looking at when you increase the levels of cell-free DNA into that and how hard it is to digest that at the same time. So I'm wondering if you have the capacity to add other material in that and see how that impacts, because we know these clots are not totally fibrin. They do have that cellular material in there, as Ed said. So how does that impact its ability to break off when you've got other processes going on?
Yeah, those are both really great questions. So adding in the effects of cells, of red blood cells in particular, and then of platelets and their contractile effects is something that we would like to do both in terms of experiments and in terms of the model going forward. And then in terms of adding cell-free DNA and how that would affect, I guess, both the mechanical stability and the enzymatic degradation of the fibrin networks is not something that we've been actively working on, but I think it's a really great suggestion that would be something that to me would be of interest, because I think it will affect both of those processes. Right.
Yeah, and certainly probably has a role in multiple diseases. I think, yes. We've heard talks today about oncological thromboses. We know that we can measure tumor-based cell-free DNA there. Certainly in sepsis, we measure cell-free DNA. All of those have risks for thromboembolic disease. So how that adds to that solidity or the ability to break up afterwards.
Yeah, no, that's definitely a great suggestion of something to look at going forward.
Others? Or if not, thank you very much, Valerie. That was a fantastic way to almost wrap up the day. And we really appreciate you coming. So we just have a couple of, before we leave, and don't leave, please. No, not yet. Turn that off. Jesse or Caleb first. First, I want to thank everybody on behalf of all the organizers and the student presentations and Valerie's presentation. Obviously fantastic. There's people behind the scenes. Caleb and Jesse have organized this from really the get-go, and the students have really been engaged in doing this. And we welcome input on how to improve this, change this. We're totally, I think we're all totally open on how to take advantage, and obviously things may change depending on whether we're in person or virtual, but don't hesitate to contact any of us and offer your your suggestions or comments. Second, Caleb or Jesse, do you guys want to take a picture of the students' presentations, the presenters? Before, if I give the awards now, they'll all disappear.
That's a great screenshot. If
Yep, a screenshot. Can we do
if
that?
you guys could put your
But you have to put
cameras
your
on,
pictures on. Valerie, you too. Don't disappear.
that would be great.
Who else? Certainly yeah, if we get everybody in the screen, then you can juggle. I don't have to be in this. But Jesse, you should. So do we have all the speakers? Make sure you're in the screen and your head isn't cut off.
Perfect. Thanks, everyone. Great. We'll get Caleb to tweet that from our account.
Great.
But I really do appreciate everyone's engagement and participation today. As Ed said, our first one, so we're open to feedback.
Okay, and now the winners. Drum roll. Okay, third place, Jaskarat Aurora. Where is she?
Thank you.
Second place, Erblin Canny. Where are you, Erblin?
Thank
Where
you.
are you? And in first place, Lisa Yamura. Fantastic, everybody. But all of them were great. I was told that the scoring was extraordinarily close. So these things are always a little tricky. But you guys all did fantastic. I was very impressed. I'm sure everybody was impressed. Okay, so with that, unless Jesse has any parting words, we can go.
That's it. I'll follow up with the trainees about the awards, and thank you
Okay,
again, everyone,
great.
for your participation today.
Great.
Thank you,
Have a
Dr.
terrific
Conway.
weekend. Okay, my pleasure. It was a joy. All right. Bye, everybody.
Hi,
everyone.
thank you.